Thank you very much for your kind introduction and also a warm welcome from my side. The following presentation will be given from my colleague, Katharina Weidmann, and myself. So I will start with the first part, and the topic is small volume packaging notified body perspective on CE marked medical devices versus Article 117 MDR combination products. As you know already, I have to present this disclaimer. The presentation reflects my understanding and is not binding. So part one from my presentation will deal with notified body perspective on Article 117 combination products. First of all, I would like to share with you notified body opinion, scope and content. Afterwards, I will focus on first experiences out of our first assessments. And afterwards, I would like to give you a short conclusions and outlook on this topic. So what combination products are we talking about in the next 10 to 15 minutes? These are combination products falling under the Medicinal Product Directive. And these can be divided at least in two major product groups. So we have the devices intended to administer medicinal product as an integral part. Here you can imagine as the most common products or devices, a prefilled syringe or insulin pens. The second major product groups are devices incorporating a medicinal substance, whereas the medicinal substance has principal mode of action, both covered in first place from the Medicinal Products Directive and also Article 117 MDR with coming into force of the MDR May last year. So here we have the legal text of the MDR. Here it is stated in the Article 1, subject matter and scope, as introduced before, the major product groups. One are with the integral part of a medicine or a drug, and the other product group are the devices intended to administer a medicinal product. So I think very interesting for you now is what are the key aspects of this Article 117. What are your kind of to-dos? So first of all, the self-declaration option was withdrawn. This is an important message for you as manufacturers. And what is Article 117? So within Article 117, you have to show the conformity of the device part, very important, not of the combination only of the device part, to the relevant general safety and performance requirements laid down in MDR. So the notified body opinion is shows the conformity to the relevant GSPRs. This is one option you can fulfill with Article 117. The other one is a declaration of conformity. And the third option is to gain or apply for an EU certificate. And this point of decision, when a notified body involvement becomes mandatory, can be performed by the classification of the device part. It's a little bit tricky. I will come to that a little bit later on in more detail. And what should the notified body opinion documentation look like, or what should be included? So we have three major parts. The core element is the GSPR documentation, ideally in the format of checklist. Second, of course, the description of the device. And the third part, instruction for use or package insert of the integral product. Very important is to keep in mind, you have to identify if a GSPR is applicable or not, and if not, please state a justification. That's really important, as we are also assessed from our competent authority, and they are very picky on that. Then please state the methods you use to demonstrate the conformity, and as well as the legal background for the methods to show conformity, like harmonized standards, common specifications, or any other guidance or applied state-of-the-art solutions. So what makes life easier for you and for us as well is the clear identification of these evidence documents and, of course, the traceability of the evidence. Let's have a look now what is the biocompatibility aspects within Article 117 assessment. They're very different to those of a conformity assessment procedure when performing a full technical documentation, including Annex 2 and Annex 3. So here within Article 117, we are dealing exclusively focusing on the compliance of the device part with the relevant GSPRs, more or less starting with 10.1 and the following. Very important, of course, is GSPR 10.3. Here we are dealing or assessing the risk of leaching and or extracting effects that affects the device performance and or usability. Or GSPR 10.4.1. Here it's a general assessment whether the drug influences the performance of the device part. A very crucial point are the CRM substances. These are stated in GSPR 10.4.2. And for example, we have here some cobalt attached to the needle. And of course, a justification is required by a concentration above 0.1 weight per weight percent can be accepted. Of course, it's depending on the user populations. Are we talking about children, adults, and so on? So there must be a justification stated why this can be accepted. One reason can also be the short contact duration. So very important is we will focus within this assessment on the biocompatibility of the single components of the device. And this is why we have in most cases, we have the justification that we can accept that the assembly does not influence the biocompatibility of the components itself and therefore not of the final device. If there are no additional substances added, like a glue or something like that. When we have a look on an example, for example, a pen injector, this can be delivered in two types. We have a needle that is CE marked. Then, of course, biocompatibility is covered by a conformity assessment procedure via the technical documentation. No need for us to assess it in more detail. And the other option is we have a staked needle. Then, of course, biocompatibility data have to be provided from the manufacturer or the pharma company or the supplier. So let's give you some insights on common pitfalls that we have identified in the past notified body opinion assessments. As mentioned before, the applicability of GSPRs. Please state a justification why it is not applicable. This is often missing, and I think this is a mistake that must not be, and it's really time consuming. Another point is the file completeness. In best case, we get a full file submission and we have one documentation package, including applicable evidence document, and we have a content, we have an overview and a link to the methods for the conformity. Next point is shelf life data. So for us, it's pretty clear that real-time aging studies are mostly not available at start of the assessment. So if we have accelerated aging data, then this can be accepted in most of the cases. But if we then will have a change to the device part and assess this device again, then of course we need this real-time aging studies to be assessed. Unfortunately, there's often missing objective evidence. This means that the GSPR checklist does not reference the corresponding documents. Only summary reports are delivered and without providing the corresponding evidence methods. Another topic is, of course, it's a lesson that all of us have to learn because of this difference between the pharma and medical device documentation and submission packages. So there's often a complex structure of submitted documentation package. So evidence documents are often difficult to identify when we get a huge document from 80 pages and we have to search for this document, for the evidence document. And statement of the supplier is another point. Without providing the reference objective evidence, it's not possible for us to assess this. So we are talking here about these CRM substances or nanomaterials or something like that. So what is the feedback from our manufacturers on notified body opinion assessments? Of course, the discussion was ongoing. Clinical evaluation board, is it needed or not? So it's not needed in any case. You have to show the compliance with GSPRs, but you can also perform this with performance data. A notified body opinion template was requested. The notified body made a position paper. This was released on October 2021. So Team NB released this template. And another problem is the notified body opinion requested by EMA or not. This is what I was talking before with regard to the classification. So it's still not really unclear. We have this Q&A document from EMA, but it does not clarify in detail. So it's kind of case-to-case decision, or better discuss with EMA and not discuss with your notified body, but ask for your notified body for the opinion. Then, as mentioned, we have the different processes and requirements between notified bodies, competent authorities, and pharma. And was really positive and make us really happy. Though the positive feedback, the predictability of timelines. So we have ensured and fixed time slots, and the suit could mostly stick to the timelines. We offer technical meetings, especially the initial meeting is very much appreciated, where they can present the product and the strategy for preparing the documentation package. And what was very well appreciated as well was the good communication with the assessor during the assessment and the exchange and the possibility to ask questions. So just what I mentioned before, we have here the Q&A document, updates and clarification. Due to the time, I will not go in much more detail here, but this is a document where you can always check what is the update, what is ongoing, and here you can stick to these decisions made by EMA. So what are the uncertainties for the future? So very big discussion with the platform approach. What about when we have a device, same device, prefilled syringe with different dosing of the same drug? Must there always be performed a new assessment in full? And this is currently under discussion with EMA, and we have no feedback so far. Then a task force was established. So notified bodies are not involved so far, as I know, but it would be great to establish a project management team with all stakeholders. But I know there is something ongoing in the background. So what is really positive and good, we have moving forward in a positive direction between MDCG and MDCG Med. In the communication, there's really something going on. What could be really very challenging for the customers and us is the lack of resources at the competent authorities. Of course, the manufacturers have concerns, and we as notified body as well, especially when then talking about consultation for the normal combination products. Yeah, how to solve this? So I think best case could be to involve all the stakeholders in the process and perhaps perform a holistic risk-based approach. Establish finally a legal framework to facilitate communication and collaboration of all stakeholders involved in the kind of assessment. So just to give you a take-home message from the RAPS convergence, here it was identified that we have the complex EU landscapes with multiple stakeholders, and we need to anticipate the notified body conformity assessment so that this is available for the marketing applicant authorization. And yeah, we need to continue developing a collaborative approach across EMA, national competent authorities, EU Commission, notified bodies, and industry. I think this was really a positive outcome from the RAPS conference last year. This is an overview of our process, how we deal with an Article 117 assessment. If you are interested in this, please let me know and come up to me, write me an email or contact me, then I can explain it to you in detail. So what is my take-home message for today? So please check your product portfolio for Article 117 applicability. In case of doubts, contact EMA. Prepare your notified body opinion documentation for the device part of your combination product. Get in touch with your notified body as soon as possible. And some add-ons. So address open points to EMA, competent authorities. Introduce your regulatory strategy to your notified body. Involve medical device experts in the early stage of design and development, and establish a GSPR list. This is the core element of your documentation. So thank you very much for listening. And if you have any questions, please don't hesitate to contact me. And now I would like to hand over to Katharina Weidmann, who will take over the part 2, the notified body perspective on CE marked medical devices.
Okay, thank you, Christiana. In my part of the presentation, I would like to explain a little bit on the impact of packaging materials on biocompatibility of medical devices and what needs to be considered in the submission to the notified body in case of a medical device product. As shown here, medical devices can be very different products. The influence and depth of evaluation needed regarding the packaging material obviously depends on the device type and the kind of packaging material used. Usually, a solid device is less likely to interact with the packaging materials than a device composed of a semi-solid or liquid material. First, let's have a look on the potential impacts on the biological safety of medical devices in general. There are a lot of factors that can affect the biological safety of a medical device during its life cycles. So first of all, of course, the raw materials. Then during manufacturing, there are impacts on the biological safety, for example, residuals from cleaning of the mixing equipment, which would be of importance. Once the medical device is in its final composition, we have the packaging materials that come in contact with the medical device, and by this potential impacting the biocompatibility. Furthermore, sterilization, transport, storage, as well as aging might impact the biological safety of a medical device. So in terms of the biocompatibility over the complete life cycle of a medical device, which needs to be shown according to ISO 10993-1 4.7, basically we have 3 different or relevant time points to be considered. So this would be manufacturing, packaging, and sterilization until we have the final medical device that is usually used for chemical and biological testing. This we would call T0. Then we have transport and storage until the end of shelf life, which would be T1. And then also until the end of the intended use, which we would call T2. T0 considers the impact of manufacturing process, packaging, and sterilization. Here, usually the following aspects are addressed: the raw materials, the processing aids, the cleaning agents or contaminations, surface treatments, transfer of contaminants, the transfer and migration from glue or ink or label through the packaging materials, the transfer from of migrants from the packaging materials, and material alterations due to sterilization conditions. Usually, an endpoint-specific risk assessment according to ISO 10993-1 is performed based on chemical and biological data. However, in addition to that, also T1 needs to be considered in the biological risk assessment. So T1 would be the end of shelf life and considers the impact of transport and storage. In addition to T0, the following aspects would have to be addressed here. The transfer of packaging contaminants, the transfer of packaging migrants, and material alterations due to storage and transport conditions. For example, the reaction of substances or degradation or corrosion. So at T1, the potential impact of packaging materials on the physical, chemical, or biological properties must be evaluated, considering the materials of the device as well as the primary packaging materials being in direct or indirect contact with the medical device to be assessed. Usually, a solid device is less likely to interact with the packaging materials than a device composed of a semi-solid or liquid material. To address the impact of the packaging materials on the biological safety of the device at T1, material data from the packaging materials can be helpful. However, according to 10993-1 6.2, USP testing performed with the packaging materials are usually not acceptable to address this risk due to the following reasons. The testing is typically conducted on raw materials rather than on the final product. The extraction conditions typically do not represent the whole shelf life, and potential interactions with the device are not addressed. So the worst case with regard to potential leachables from primary packaging materials would be a liquid device where leaching basically takes place during the complete shelf life. Examples would be wettening eye drops in a blow-fill seal container or a dermal filler in a pre-filled syringe. So here you can see an example of a pre-filled syringe. Here we have different substances inside of the different packaging materials used for the device. So this pre-filled syringe consists of several components like the tip cap, the plunger stopper, and the syringe barrel. And all these materials have contact to the medical device. So during shelf life, which could be, for example, 2 years of storage at room temperature, some of these substances might migrate from the packaging materials into the medical device itself. Maybe also from the label or its glue, substances might migrate through the primary packaging materials into the device. By this, these substances potentially can impact the biological safety of the device. In this case, probably theoretical assumptions based on material data might not be sufficient to address the potential toxicological risk. On the other hand, chemical analysis of this kind of devices after accelerated or real-time aging is often technically very challenging, as high concentrations of substances intentionally contained in the medical device might mask leachables in low concentrations during the analysis. Instead, chemical analytical testing and toxicological risk assessment of the packaging materials can be performed in order to determine the potential leachables during shelf life and address their toxicological concern with regard to the biological safety of the medical device. So for such analysis, one very critical aspect are the chosen extraction conditions. They are crucial for the representativeness of the results. So the extraction conditions shall be documented and justified. They shall be relevant for the conditions during shelf life. And the choice of the test sample is very critical because an unfilled syringe or a syringe filled with extraction medium already during manufacturing should always be preferred rather than a medical device manufactured as in routine and then emptied for analysis of the extraction container, as here already extractables or leachables might get lost. To address a complete shelf life, usually accelerated or real-time aging in the container of interest or exhaustive extraction would be necessary. For exhaustive extraction, several extraction steps might be necessary until the extracted material is less than 10% of the initially extracted amount of material. And by this, the maximum amount of extractables is reached that can be released from the material under assessment, and a toxicological risk assessment of those is considered to assume the worst case. Another critical factor would be the selection of the analytical methods, which should be able to detect the substances that are expected, as well as possibly unknown substances in toxicologically relevant concentrations. It should be validated, and they should have appropriate sensitivity, which should be considered in the toxicological risk assessment. So if there are any questions, I will be happy to answer them in the Q&A session. Thank you for your attention.